Okay, well, first of all, hello everybody. Well, my name is Antonio Borlan Miñarro, and I'm come from Spain. And well, I'm going to upload the presentation. Okay, can you watch it?
Yes, we can see your screen. Please start.
Okay. Well, so first of all, thanks to IGSMP for the invitation, for counting on me. Well, my name is Antonio Borlan Miñarro, and I am biotechnologist and computational biologist. I come from Spain, and I'm going to do a presentation which is called The Era of Bioinformatics, Multi-omics Analysis and Machine Learning Methods Applied to Health Issues. So here are the contents of the presentation. And as it's the first of the webinar, I'm going to make a little introduction about biotechnology and bioinformatics, so as to have a little background in the field, in this topic. So, well, biotechnology is a relative novel science, but it has existed since the beginning of civilization with the domestication of plants and animals. But the definition is the use of biology so as to obtain products, improvements, or in general goods to improve the human health and society. And it's a quite wide field, and it's made up by a lot of different areas such as molecular biology, chemistry, genomics, genetic engineering, nanotechnology, or even informatics. Biotechnology itself is classified in many different branches that are classified by colors. For instance, red color is medical and pharmaceutical biotechnology. Green one is related to agriculture, yellow to food production, and the one in which we are going to focus on in this webinar, which is gold, related to bioinformatics and data analysis. So let's move on to the bioinformatics field. Well, bioinformatics, but let's start with the sequencing technologies, which many years for over the years in 1980, we can see here that the kilobases of nucleotides that could be sequenced by day by machine were really, really low. So it was inefficient, and the cost per kilobase was quite high. And over the years, the yield in these costs per kilobase sequenced has grown exponentially to the point that today we can obtain more than 1 billion of kilobases per day per machine. So it's a quite huge number, and it has been really fast. So now we need the necessary technology, the necessary tool, so as to analyze this information. Here is where the bioinformatics arises from, which is basically the analysis of data so as to obtain information and knowledge. There are also a lot of branches in bioinformatics, and here I'm going to show you just some of them. One of them could be genomic data analysis, for instance, comparative genomics between species, so as to compare the genome, the transcriptome, or even the proteins among species. Also, single cell RNA sequencing and analysis, and spatial transcriptomics. This last that I have said, single cell, allow us to have the transcriptome of a unique cell type, and a spatial transcriptomic allows us to know the location of this cell in the human body or wherever. So this is a really potential tool that it's being used a lot nowadays. Another branch is structural biology. For instance, we can know about the molecular structure and dynamics of macromolecules, for instance, proteins, and it's really helpful in the field of drug design and discovery because maybe 10, 20 years ago, it was a quite tedious, task to design a new drug because it was done from scratch. And right now, we can take some molecules that we already know and that are similar to others, so as to obtain in an easy way the drug for a target. Well, another branch, the evolutionary biology, with the comparison of species or the simulation of the evolution of population with the offspring or even the generation. We can simulate with code, basically. And the machine learning branch, which is the use of artificial intelligence to predict behavior. Here we can distinguish between two types of machine learning models, which are supervised and unsupervised. Supervised are related to those in which we know the input and the output. I mean, for instance, we have a patient with some features and we know if he has or not a disease. And the unsupervised models, we just know the input. So we don't know the disease, but these are related more to classify into groups, maybe the type of the stage of a disease itself. So now let's move on to the practical examples that are based on my bachelor and my master thesis. First of them is a multi-omics analysis. And to have a context, the main aim of this project was to determine omic differences between two groups, which were mothers of term newborns, here we have, and mothers of preterm newborns who were born before they had to. So breast milk was extracted and the exosomes of this breast milk were analyzed so as to obtain microRNAs and lipids. So two different analyses were carried out, which was the transcriptomic analysis and lipidomic analysis, so as to obtain the differences between these two groups. Let's start with the transcriptomic analysis. First of all, we received the sequencing reads, which are a lot of nucleotide reads of more or less 20 nucleotides, because that's the length of microRNAs. And the objective of this step, the preprocessing step, is to obtain an expression matrix in which we have the counts of every single microRNA in every patient. So the first step is to do a quality control of the sequence. After that, the trimming, which is to remove the adaptators or primers, which are source sequences that we add in both sides of the sequences of the microRNA in order to sequence them. After that, a post-quality control, so as to check it out if the trimming was done correctly. Then when we have the sequences itself raw without the adaptators, we have to map, in this case, to the transcriptome, the human transcriptome, because this is RNA. And To map, it means to align our sequences to the transcriptome so as to obtain to which microRNA these sequences belong to. After that, another mapping quality filter, and then we have the expression quantification in which after that we have this expression matrix in which we have the counts. So now it's time to analyze or to obtain which microRNAs are overexpressed or underexpressed in one group in comparison with other. So in the case of microRNAs, we found in every group 20x microRNAs which were overexpressed. And in the case of lipids, there wasn't a preprocessing step. There was unnecessary because we already have from the mass spectrometry a matrix with the counts of every lipid. So we could do the analysis of concentration directly. So we obtained that 21 of the protein group were in higher concentration significantly in comparison with term, and 9 in the case of term. So when we have all the microRNAs and lipids that are found in more concentration in one group than in other, in this case, this is for the lipids, we did an enrichment analysis so as to obtain, well, in which group, what were the lipids that were in higher concentration and the pathways, the biological pathways in which they were involved. So in this case, in the lipids found in higher concentration in preterm group, we found that they were mainly involved in cell signaling and energy storage. And in lipids with high concentration in term group, they were mainly involved in pro-apoptotic effects. On the other hand, in the case of transcriptomic data, we had the differential expressed microRNA, so the enrichment analysis was focused to obtain the target genes that of these microRNAs, and when we have the target genes, we obtain the biological pathways in which these target genes were involved. So he has the final table in which we have the top 10 Go terms that are the most relevant. And here we can see that the apoptotic process, this is the same that we found in the other case in the lipidomics analysis. So then the final step of the study was to integrate both information, lipidomic and transcriptomics, so as to obtain a biological meaning. So we have to study, in this case, the apoptosis process, and to study in a way that in the newborn subject that we were studying. So to obtain a biological meaning and a conclusion of the differences between both groups. So let's move now to the second example, which is machine learning applied to CVD risk. CVD is a cardiovascular disease. And well, to put you in context, there are already some calculators, which are scales, for instance, ASCVD, Framingham, or Hertz score. These are the three traditional ones that can calculate the risk that you have to suffer from a CVD in a range of 10 years, just with your demographic features such as gender and age, and some risk factors that this change among them. For instance, we can see cholesterol levels, blood pressure, diabetes, smoking habits, etc. So you introduce your values, and then you obtain the risk that you have of suffering this CVD in 10 years in a percentage. For instance, in this case, 6.9%. But what's the problem in here? That these calculators just only take into account demographic data and risk factors. But there is a quite important risk factor that hasn't been used in these calculators, which is the plaque data. Plaque is the fat that you find inside your blood vessels, and it's known as a reliable biomarker of this disease. So that's the purpose of this project, that was to carry out a model of prediction in which we obtain the risk of suffering a CVD, taking into account demographic data, risk factor, and also plaque data. So here we have the project pipeline, in which the first step was the data collection, apart from clinical reports. After that, the preprocessing of all the data, the creation of different data sets, every one of them with different features in order to analyze the results, their relation with the risk, different machine learning algorithms, classification models, and regression models. And after that, the presentation and visualization of results so as to obtain different conclusions. Here, it's the machine learning model schema, in which the first step was to obtain all the features, which, as I have said, sorry, were demographic features, age and sex, risk scales, which are the calculators. So we obtain the values of risk of every patient, risk factors, which were the use of these scales and many others, and plaque data, which is the novelty from 5 different territories, which are artoiliac and femorals and carotides. When we have all these features, we carry out different machine learning models. And when we have the models, we evaluate them so as to know the adaptation of plaque data to the current traditional risk scales. So in this process, the first step after the data collection is the data preprocessing. The first sub-step, we have to remove those features and patients which have a lot of missing values. In this case, it was set a threshold of 20% of missing values. After that, we have to estimate those resting missing values, because we have to have a matrix of complete of values. There cannot be a non-value. We have to transform some features, for instance, categorical features, to numerical values. And after that, the most crucial step is to normalize the data, because we need to standardize all the features to make them have the same weight when performing a model. Because if not, it's going to be unbalanced. And some features are going to be more powerful in the decision of the model, and they need to have the same weight, the same relation. Now we have all the preprocessing step finished. We have the input of our models and the output of our models. For the input, we use different data sets for different data sets, and every one of them had different features. For instance, the dataset 1, the purpose was to evaluate every feature that we had, apart from those related to scores. Dataset 2 was only plug features. Dataset 3, all features apart from risk factor used in the calculation of the traditional risk scores. And another one in which was dataset 3, but with a feature selection analysis of just the 10 most important features. And in case of the output, the values, the features used as outputs were the 3 risks calculated, the traditional risk scales, and another one additional that was made up by the combination of the other 3. And there were 4 different possible values in these scales, which was low, from 0 to 5%, borderline, intermediate, and high. So with these 4 different data sets as inputs, the 4 different outputs, which are the risk scores with 4 values, only 4 values, 4 possible values. Another 4 algorithms were used so as to carry out the different models, which were logistic regression classifier, decision tree classifier, random forest, and multi-layer perceptron. So within the combination of every one of them, for instance, dataset 1 with logistic regression and ASCBD, another model, dataset 1, logistic and Framingham, and with every combination, a total of 64 models were carried out so as to analyze the prediction in every case. These tables are a sum up of all the models, all the 64 models, and these are the ones that obtain the best results. And so as to evaluate the results of the models of the prediction, we had different metrics, which are here in the top row, which is precision, recall, F1 score, accuracy, and the F1 score of every risk class, which is low class. Here is 1/2 because 2 intermediate risks were merged in order to obtain better results, and F1 of high risk score, high risk class. And the metrics, in order to evaluate them, we have to take into account that if the metric is close to 1, it's a good metric, and if it's close to 0, it's a bad metric. The prediction was carried out in a bad way. So here we can see that the best model that was obtained was with the dataset 3 after a feature selection analysis as an input, ASCBD score as an output with the decision tree algorithm, and we can see that the metrics are all of them are higher than 0.9%, which means that they are really good metrics, and it has been a good prediction. So it has been everything. Thank you, all of you, for being here. It has been a pleasure. And now if you have any question or doubts, I would be glad to answer you. So thank you very much.